Welcome, everybody. Thank you for sticking it out to this final session of the meeting here, the host response to infection. My name is Matt Long. I'm assistant professor at Ohio State University. Just for housekeeping, I'm sure you're all aware of how to use the app for questions, so feel free to put them in. If that's not working for you, please just wander towards the front after the end of each talk, and you can ask your question in person. We can repeat it so the whole audience can hear. So without any delay, I'd like to introduce Clemente Brito, assistant professor at Yale, who's gonna take you through an intro.
All right, well, hey, Saturday Warriors, you finally made it to the very, very end. So thank you so much for being here and sharing this session with us on host response to infection. These are old, after this conference, these slides are old, but I think it's important just to draw attention to the fact that Trikafta and modulators in general have transformed the landscape of how we see microbiology in our patients and how we see the interaction between those hosts and their pathogens and how that clinical course evolves. We have been learning more and more through this conference about how these interactions shape the future decisions about therapy and how they shape the progression and the clinical course of our patients. In fact, just yesterday, we learned that Simplify did not find any difference between continuing hypertonic saline and pulmozyme, for example. And this is, I think, to a certain degree, a reflection of how far we have come. So specifically, more germane to what we're going to be talking about today, we've seen significant differences in the microbiome and in the microbial landscape of our patients as we treat them with modulators in the past few years. And these are just some examples from an article that I pulled in which you can see how different our sputum, the predominant pathogens and other commensals are in one sputum versus another. And just without going through all these different articles, what you see here is just how many reports of decreased Pseudomonas burden there are, how many reports of increased microbial diversity are available now. Oh, I realized that I just copied that twice. So I guess it's also important to point out that there may be not only direct interactions with the drugs themselves, but also interactions with the immune system that are relevant to what we're going to be talking about today. And I think it's kind of a running joke now that you cannot come to NACFC at a host pathogen lecture and not talk about Katie's article from 2017. Where are you, Katie? Yeah. So we have wonderful speakers today that have contributed dramatically to our understanding of these interactions. There's amazing work from Terry, amazing work from Callie's lab with Goshun that has done incredible work with neutrophil pathogenesis as well. So we're going to try to bring down some of our understanding from this and previous sessions into these questions that remain unanswered, which are, for example, what is the role of polymicrobial infections in the CF and innate and adaptive immune response? Mechanisms that connect directly microbiota changes with host defense, and how age and sex differences can actually contribute to this modulator response and its immune effects. So we're going to work on defining host and pathogen feedback mechanisms. We're going to discuss some of these specific cell subsets and how they impact the host response to pulmonary bacterial infections, and finally discuss some of the relationships between CFTR dysfunction and the airway and intestinal microbiota. So these are our outstanding speakers for today. Terry from Lurie Children's, who will be speaking about relationships between inflammation, bacterial ecology, and the BAL from CF Airway. Callie, coming from New Orleans, she's going to talk to us about epithelial and immune defects and how they influence intestinal microbiota. Sebastian from Colombia will be talking to us about pulmonary ketogenesis and how that influences Pseudomonas tolerance in the CF lung. Nicola from the Ospedale Centro San Raffaele in Milan. So he's going to talk to us about the role of IL-17 receptor C and in the pathogenesis of infections with Pseudomonas. And finally, from Yale, Hassan Oz, will be talking to us about a unique subset of CCR2 monocytes and how they are involved in host defense against Pseudomonas. So having introduced all of this, I hope you share my enthusiasm in bringing them up to the podium. I hope you will be very active and very involved, and we'll try to get a couple of questions in halfway, through each talk. But then at the end, whatever's on your mind, please share it. We want to hear it, and we want to try to use this to push the field forward as much as we can. And that's it. Welcome.
All right, well, our first speaker today is Dr. Teresa Laguna.
Okay, awesome, thanks. All right, thank you for the invitation to speak to you guys. I'm excited to actually talk about some research, which I haven't gotten an opportunity to talk about in quite some time. So I don't have any disclosures that are relevant to the talk today. I do want to start with an important message that I hope you guys have already seen throughout this conference. For the safety of our presenters and attendees, I urge the Cystic Fibrosis Foundation to commit to ensuring that future meetings are held in locations where elected leaders understand that healthcare is a human right and affirm the importance of reproductive freedom, gender-affirming care, anti-racist actions, voting rights, and the sanctity of the physician-patient relationship. Awesome, thanks for your support. So, progressive lung disease secondary to chronic airway infection and inflammation is the leading cause of morbidity and mortality in people with CF. We've all seen this schematic over and over again about how defective CFTR ends with respiratory failure and ultimately death. Embedded in this process is this cycle of mucus obstruction, infection, and inflammation. That while a highly effective modulator therapy has improved FEV1, facilitated growth, and decreased pulmonary exacerbations, we do know that infection and inflammation in some way, shape, or form continues to exist. We know from human and animal models that inflammation, neutrophil elastase, and IL-8 can be present in the airways in the absence of infection and impact airway health. How we've thought about infection in the CF airway has really been defined by typical respiratory bacterial pathogens that we identify through a culture of airway secretions, whether it's an OP swab, an expectorated or induced sputum sample, or lavage fluid. And these are the typical bacteria that we've talked for years about: Staph, Haemophilus, Pseudomonas, Burkholderia, Achromobacter, and Stenotrophomonas. However, with next-generation sequencing of airway secretions, we really have broadened our view of infection with the identification of complex and polymicrobial communities that exist in the CF airway, both the upper and lower airways of children with and without CF. So we do know that infection and inflammation are intertwined in some way. However, there still is much to learn, and there's an opportunity to determine whether there are independent drivers of each to help us take better care of our patients and guide therapeutic development. So the objective of our study was really to integrate our 16S bacterial sequencing data with measured concentrations of inflammatory cell counts, cytokines, and airway proteases such as neutrophil elastase and cathepsin B from bronchoalveolar lavage fluid samples from children with and without CF to try to begin to investigate the dynamic relationship and complex interplay between infection and inflammation. So we used banked BALF specimens that had been collected nationwide from clinically indicated bronchoscopies, so these were not stable children, performed on kids with and without CF, and we collected the fluid along with relevant demographic and clinical information. We did quantitative polymerase chain reaction to measure total bacterial load and 16S rRNA sequencing to characterize the bacterial communities. And we did this in collaboration with the Cystic Fibrosis Microbiome Analysis Core at the University of Colorado. We measured a cytokine panel, the members listed here, in BALF in collaboration with the CTRC, Center for Biochemical Markers at the University of Colorado in Denver. And then we measured neutrophil elastase and cathepsin B in BALF using a laboratory-developed spectrophometric assay and ELISA, respectively, in my laboratory. The statistical analysis was performed by Dr. Brandy Wagner, also at the University of Colorado. Inflammatory measures were compared across disease groups using a Wilcoxon sign rank test, and then Fisher's exact test was used to compare the detection across groups. We used the Shannon index to measure microbial diversity, and then we did a hierarchical clustering analysis on the 16S rRNA data, which included annotation with inflammatory, microbial, and relevant clinical measures. That sounds like a mouthful. Once we get to the actual data, I think it'll start to make a little bit more sense. And hopefully Brandy's in the audience to answer questions. Okay, so in terms of results, so this is a typical table 1, just a real high-level view of the subjects that we studied in our study. So we had 51 CF patients and 73 disease controls. These are individual BALF samples from each. Our average age was somewhere in the neighborhood of 6, so these were kids. And as you can imagine, the CF patients were all had a high amount of neutrophils in their lavage fluid. The disease controls did not. It's worth mentioning that our disease control subjects, just to define them for you guys, was really any pediatric patient who did not have CF, who underwent a clinically indicated bronchoscopy. So this was a very diverse group of kids with cancer, kids with rheumatologic conditions, kids who are immunosuppressed, really undergoing a lavage sample for rule-out infection. So a wide variety of kids were all in this group. So I'm going to run through a few of the results that we have. So this is taken directly from a paper that's been previously published in late 2021. I encourage you to go to the paper for additional details. This was the results of our sequencing within CF BALF of CF and non-CF BALF. So what you're looking at here is a heat map which shows relative abundance of specific taxa across age. So it's a heat map of sequencing results for all BALF samples in CF and disease control groups, arranged in order of increasing age. So across the bottom, across the x-axis, you have relative abundance going from low to high. The samples are all also ordered by age. And on the y-axis, you have all the different taxa that needed to be present in greater than 40% relative abundance or more to be included. So what you see on the CF side are the typical pathogens that you would expect. So Stenotrophomonas was represented only on the CF samples. If you look on the control side, none of them had Stenotrophomonas. Pseudomonas aeruginosa, again, predominantly represented in the CF samples at a later age. Again, not much on the control side. Haemophilus influenza, which again, in the CF samples was present, but particularly in an early age, which is what we see clinically. And on the control side, present throughout the age spectrum. And then Staphylococcus aureus, which you really see present throughout almost all the BALF samples. On the control side, you really see Streptococcus mitis group present throughout on the control side, which you really didn't see in the CF samples. And then Moraxella, which again, not really on the CF side, but you see it on the control side. So results here, there are clear distinctions in microbiota across the age spectrum. And there are differences between CF patients and non-CF patients, regardless of what the non-CF patient's underlying condition was. Again, for further details, please go to the paper. So then what about the other things that we did? So as expected, neutrophil concentration is much higher in CF BALF compared to the disease controls. So this figure on the x-axis is showing you CF versus disease controls, and on the y-axis is the log of neutrophil elastase. So what you see is that neutrophil elastase concentrations were significantly higher in CF subjects compared to disease controls, not unexpected given the degree of neutrophilic inflammation. Most neutrophil elastase measurements were in fact below the limit of detection in the disease control sample, so they didn't really have much of it present. So we actually had to impute those values with the lower limit of detection to be able to visualize it on the figure. But they really didn't have much, any at all, in their BALF. In terms of our cytokines, again, kind of what you would expect. Across the y-axis is the panel of cytokines. Across the x-axis is the panel of cytokines. Across the y is the log concentration of the various cytokines. And Again, what you would expect, concentrations of IL-1 beta and IL-8 are significantly higher in CF BALF compared to disease controls. So cathepsin B. So some of you may or may not be familiar with cathepsin B. So I'll just give you a little bit of background before I present those results. So it's actually an elastolytic cysteine protease that's linked to remodeling of extracellular matrix, bacterial infection, and biofilm formation in the CF airway, specifically involving Pseudomonas aeruginosa. So it's been reported before, and my lab has actually done previous work in cathepsin B. So again, I refer you to a paper for more details. We actually did this in little kids. So we showed higher concentrations of cathepsin B in the urine of CF infants compared to healthy controls, looking for biomarkers that you could obtain non-invasively from a little baby. And urine is the easiest one to get. So at the time, we proposed it to be a biomarker of lung disease in CF, but hadn't really had an opportunity to really study it further until we got this nice sample set of lavage fluid samples. So we decided to take advantage of that. So what did we find? So we found that cathepsin B is much higher in CF lavage fluid samples. So again, across the x-axis, you have age, and this time we broke it up into groups. So less than 2, 2 to 5, you can read the rest across the bottom. On the y-axis is concentration of cathepsin B in the lavage fluid. Blue is disease control, red is cystic fibrosis. So what you see is cathepsin B concentrations in CF BALF across the board were significantly higher compared to the disease controls. The median values were significantly different, with CF having 13.5 nanograms per mil compared to the disease controls of 3.6. And what was interesting was that cathepsin B concentration was notably higher in the 0 to 5-year-old range in children with CF. As a pediatric pulmonologist that takes care of CF kids, we worry about that initial window, that early window of 2 to 5, where we know they're infected, we know they're inflamed, but we can't do PFTs, they don't make sputum, and so it's really hard for us to really know what's going on. So that's been our particular interest, is really looking in those little kids. Okay, so I know this is busy, but this is a really cool slide. So give me a second, I'll walk you through what we're looking at. There's a lot of information, but a lot of cool things to think about here. Okay, so this is another heat map of relative abundances of traditional CF pathogens and anaerobic bacteria associated with CF. So I'll walk you through what you're looking at. Okay, so on the y-axis, you have typical CF pathogens. So we chose staph, strep, Stenotrophomonas, Pseudomonas as our representatives from the CF side. And then we have some anaerobes that are also typically seen in the CF airway. So yeah, Prevotella, Streptococcus. We included Haemophilus, Veillonella, and Fusobacterium. Across the x-axis, you have individual BALF samples. So each individual bar, which will make sense in a second, is an actual BALF sample. Red is for CF, blue is for disease controls. Again, across the y-axis, you see all the individual clinical markers. So I have an arrow pointing at IL-8. There's cathepsin B. There's neutrophil elastase and percent neutrophils. Shannon diversity, which is just a measure of how diverse, how many bacterial communities are actually within a sample. Lung function, and then at the very bottom is age. All these different colors are shaded so you know the concentration or the amount. So the darker the color, the more that is present, or the higher the number. So lots of things to look at here, but I'm going to direct you to a couple things. So the first is this staph cluster. So well, first let me explain what we did. How about that? So basically what we did was we did a clustering technique, which is actually pretty cool. So the BALF samples were clustered based on a diversity measure that we call Morishita horn. So basically how similar are samples together. And that's how they were clustered. The red and blue across the top is really how close these samples were clustered. How much alike are they? So that was really the first thing that we did. Oops. And then samples were annotated with measures of inflammation as well as clinical measures. So when you look at this Staphylococcus dark bar, you can follow it up to see what individual samples had what. If I had that, there we go. So here's our staph cluster, and if you follow it up, you can tell that there's dark bars here, there's some dark bars here. So this Shannon diversity amount is pretty light. So these are samples that were predominantly staph. They weren't very diverse. They were mostly staph that were in there. And then if you continue to follow it up, you'll notice that there's a lot of neutrophils, a lot of neutrophil elastase, a lot of cathepsin B, and a lot of IL-8 in the staph cluster. That's interesting because when you compare it to the Pseudomonas cluster, so I have that one highlighted now, and you do the same exercise and follow it straight up, you see the same kind of story where you have a less Shannon diversity. So again, these samples are predominantly Pseudomonas. You have a lot of percent neutrophils, a lot of neutrophil elastase, a lot of cathepsin B. I was surprised, honestly, about this because I don't think of staph as really inciting that degree of inflammation in the airway. Again, as a pediatric lung doc, we traditionally don't treat staph when we see it in an airway culture of a child. We treat Pseudomonas, but we don't treat staph. So this was interesting to see that this cluster, this staph, seemed to generate as much of an inflammatory response as Pseudomonas. And the last thing to point out is this other big cluster here, which is mostly Prevotella and Streptococcus. These were seen in little kids, so the 2 to 5 age group, and did not incite a lot of inflammation. So there was less neutrophils, less neutrophil elastase, less cathepsin B. So again, a lot to think about, but a lot of really cool associations here that I was a little bit surprised to see. So in conclusion, children with CF exhibit distinct airway inflammatory profiles compared to disease control subjects without CF, characterized by increased cytokines, white blood cells, neutrophils, and airway proteases. Cathepsin B is significantly higher in CF BALF compared to disease controls and is higher early in life, suggesting an early inflammatory process. And while additional analysis are required, it's possible that staph may elicit a very particular immune response early in life that may be critical to the development and progression of lung disease. So what are we going to do next? So obviously we're very interested in the staph cluster. There's a story of heightened inflammation in the setting of early age and higher lung function that has a potential to have an impact on the development and progression of what comes next. And then we need to better understand cathepsin B. We also have done a lot more work with the same BALF sample set. So we have metabolomics data, metaproteomics data. So we're going to be integrating all of this into a multi-omic analysis to really try to understand what is actively going on within the lower airways of kids with and without CF. It's an important control group to be able to compare it to. Whoops. So lots of people to thank, especially the patients and families who volunteer to be in studies like this. So we're very grateful for that. Happy to take questions. Thanks.
Thank you, Terry. That was really fascinating. Any questions from the audience? Do we have All right. So we have a question from Katie Heisert for Terry. How many subjects were co-infected? Did many have staph and Pseudomonas?
Yeah, so the analysis that we did was really looking for them individually. But you're right in that when they're co-infected, it has a big impact. What we were trying to determine was what might be the impact of very specific individual pathogens. So yes, we have done that. That will come in the paper that's soon to be out. Yeah,
And
important
then,
point.
We got another question from Katie Boyne asking if you know if the staph cluster was predominantly MSSA or MRSA.
Yeah, it's a good question. So we don't have the capability of telling the difference on sequencing, but we can tell with culture, and I can tell you that they were mostly MSSA. Good question.
Can I ask a question?
Of course you can.
So it looks like, so your work is on really young patients, right?
Yes.
So these are the people that we hope in the future will not have a lot of structural disease and will behave differently. But what could you infer a little bit from what you know about what these data would look like in our middle-aged 40s and 50-year-olds, say age, how that has affected, how would that be different from this?
Yeah, well, I mean, I think the older you get, the less diverse your samples are. So, I think one of the biggest take-home points for me and what has been most enlightening about working in this area has been there seems to be a progression of the bacterial communities that inhabit the airway. And what you have as a 35 or 40-year-old is a result of what you had as a 5-year-old. And so if you can impact what happens early, in whatever way that is, you can have a dramatic impact on what occurs in an adult. Because by the time you get there, I mean, it's already done. So I think there's an important window in the early kids to really understand how and why these polymicrobial infections happen to be able to have an impact on the adults.
Right, and this is a real quick one. Were
Yeah.
they on modulators?
No, this is great question. No, this was before the modulator era, so none of them were on modulators.
I have a follow-up question. Do you know, can you speculate what you think the major source of the cathepsin B is?
Yeah.
Is it released phagocytes or epithelial?
It's a fantastic question, and it's on our list of things to do. Yeah.
All right, so thank you,
Oh,
Terry,
yeah.
so much.
Yep.
Keep thinking of those questions. We'll continue asking them at the end.
All right, our next speaker is Callie Skull.
Thank you for having me here. Let me turn this on real fast. Like they said, my name is Callie Skull. I am a PhD candidate from LSU, HSC in New Orleans, and I am in Dr. Goshan Wong's lab. So today I'll be talking about how the epithelial defect and the immune defect contribute to the intestinal microbiota. So if you were at the GI and motility talk this morning, we got a really nice reminder that cystic fibrosis initially was a gastrointestinal disease. And although now the predominant focus is the lung disease, especially in adults, it still does present as an intestinal disease in infants. And so a brief overview of the intestinal disease is that they suffer from small intestinal bacterial overgrowth, or SIBO, in which the bacterial populations in the small intestine are at higher abundance than what they should be as compared to healthy individuals. Individuals with CF also suffer from dysbiosis, in which the bacterial taxa that are present are different in CF individuals than they are when you compare them to healthy individuals. People with CF suffer from distal intestinal obstruction syndrome, which is when the intestinal contents become compacted and are not able to be passed. Many people with CF suffer from pancreatic insufficiency, which has been linked to malnutrition and malabsorption. It's also been linked to CF-related diabetes. And individuals with CF have a 10-fold increased risk of colon cancer and are actually required to get colonoscopies 10 years prior to healthy controls. There are some studies that show that there's a correlation between inflammatory bowel disease and CF, in which individuals with CF have a 7 times increased chance of getting inflammatory bowel disease and a 17 times increased chance of having Crohn's disease. But regardless of the association of CF and these inflammatory disease, the symptoms that are present are identical. And so our lab's focus is on dysbiosis, so how the alteration of the gut microbiota leads to some of the symptoms seen in the intestinal disease, and the relation of the immune system, so how this dysregulation in the intestinal disease and the immune system correlate. So honing in on the CF microbiota, there's been a number of studies that show that CFTR dysfunction in humans, mice, rabbits, and a few other CF models actively select the gut microbiota, in which these either humans or animal models have a drastically different microbiota than their healthy cohorts. A study showed that taxa is able to contribute to the increased malabsorption, suggesting that a dysbiosis is linked to the malnutrition seen in individuals with CF, as well as heightening the intestinal inflammation. Current studies use a whole body CF model, which is great because this is most translational to what is seen in humans, but it does have limitations. And lastly, there has been new studies showing that the immune system has a direct influential role on the gut microbiota, showing that there is a symbiotic relationship between the two, and when one is dysregulated, the other one gets dysregulated. So current research believes that the CF disease is caused by the epithelial defect, but we wanted to question that. So we asked, how does the immune defect contribute to the altered gut microbiota? So what we did in brief is we took 5 mice. We have a wild-type mouse with 4 CF mouse models. We co-housed them together to normalize for any environmental conditions. And then I collected the small and large intestinal contents as well as fecal pellets, and they were processed for 16S rRNA analysis. So to better understand our mouse models, this is a very simple table. Our wild type are wild type. Every cell that should express CFTR does express CFTR. Our pan-CF is a full CFTR knockout, so there's no CFTR expression in any cells. Then we have 3 immune-specific models. Our myCF, in which all myeloid lineage cells have CFTR expression knocked out. So specifically for our group, we're looking at neutrophils and macrophages, so the innate immune system. But yet the epithelial still has CFTR expression. Then we have 2 single immune knockout models, a new CF, in which the neutrophils have loss of CFTR expression, and the MACFC, in which the macrophages have loss of CFTR expression. So what we found here is that within the small intestine, our knockout models do have differing taxa when compared to wild type, but most drastically is in our pan-CF mice. And so this was good, this was exciting. It validated our mouse model because this matches what is seen in the literature. So we wanted to look at it a little bit further. So we did Shannon Index, which is an alpha diversity metric, which we heard from Dr. Terry. And in addition to measuring the diversity of the taxa, it also measures the evenness, or how proportional the taxa are to each other. And so we can see in community A that the taxa are perfectly even. In community B, there is moderate unevenness, but nothing like dysbiosis. This is pretty exemplary of what is seen in a healthy microbiota. And then in community C, this would be dysbiosis. You are predominantly colonized by a single taxa, very unhealthy. And so if we zoom in on that box plot, we see that our pan-CF in green are significantly different than our wild-type mice. That matches what's seen in the literature. Interestingly, they are also significantly different than our single immune knockouts, so our MACFC in blue and our new CF in red. But they are not significantly different than our myCF. So this indicates that when CFTR is knocked out on multiple immune cell types, that the evenness of the bacteria present is reduced, making them more similar to the taxa seen in pan-CF. This suggests that the CFTR expression on myeloid lineage cells is able to influence the intestinal microbiota. We then did a second alpha diversity measure using Faith's PD, or phylogenetic diversity, and this measures how similarly related the taxa are based off of phylogeny. And so again, zooming on this box plot, we see that our pan-CF are significantly different than wild type, and additionally significantly different than our MACFC and our new CF. But again, the most interesting finding was that our myCF were not significantly different than pan-CF. So again, this suggests that when multiple immune cells lack CFTR expression, we reduce the taxa evenness, and we also make the taxa that are present more phylogenetically similar to our pan-CF mice. So these 2 findings show that the immune system is susceptible to CFTR dysfunction on the immune cells. So the next thing we did was Bray-Curtis. This is a beta analysis that measures how similar or dissimilar your subjects are from each other. And so what we found is that the small intestine in pink is most susceptible to changes in the immune cells. So we see that when we lose CFTR expression in either our whole knockout model or in one of our immune knockout lineages, that it creates diversity within the small intestine. We see less in the large intestine and then in fecal. So what we know so far is that the loss of CFTR on immune cells affects the gut microbiota. We did use 5 different mouse lines, and so the genetic differences may be the influential factor. So what we did to alleviate this problem was to do sibling-matched bone marrow transplantation so that the mice all had the same genetic background. So in brief, we took bone marrow from either a wild-type donor or a CF donor, and then we gave it to irradiated recipients, and we created 3 groups. Group 1 and 2 allows us to compare the epithelial difference. So a wild-type bone marrow was given to a wild-type mouse. Wild-type bone marrow was then given to a CF mouse. So these mice have the same wild-type immune system, but one has a wild-type epithelium, and then one has a CF epithelium. Then comparing groups 2 and group 3, we can look at the immune difference. So we gave wild-type bone marrow or the immune system to a CF mouse, and then we can compare it to a CF mouse that was given CF bone marrow. So now they have the same epithelial genotype, but one has a wild-type genotype immune system, and then one has a CF immune system. So again, to better understand this, we can look at this table. Wild-type to wild-type will have CFTR expression in both the epithelial and the immune cells. Wild-type to CF now has a functioning CFTR expression on immune cells. And then again, our CF to CF does not have CFTR expression on anything. And so again, when looking at our Bray-Curtis PCA plot, we see that when we compare our CF to CF in yellow to our wild-type to CF, so same epithelial genotype, but now one has a functioning CFTR on their immune cells, these mice become two distinct populations when comparing the small intestinal microbiome. This shows that by altering the immune system, we are able to alter the small intestinal bacteria. So we wanted to look at these bacteria that are different. And what we found is that when comparing the immune difference, so our wild type to CF versus our CF to CF, there were 8 significantly different taxa in the small intestine. Whereas comparing the epithelial defect, in which they both have wild type immune system, but one has a wild type epithelial and one has a CF epithelial, that there was only one significantly different taxa, indicating that the immune system is mainly altered by, the small intestinal taxa is mainly altered by the immune defect. We see the opposite in the large intestine, in which the epithelial difference is able to cause 6 differences, whereas the immune difference only contributed to 5 differences. So we wanted to take this a step further and look at the predicted functions of the bacteria taxa that are present. And so by using PyCrust, which is a bioinformatics program, we are able to look at bacterial predictive functions based off of gene expression. And so you use the Kyoto Encyclopedia of Genes and Genomes, or KEG, which is a huge database for systemic analysis of gene functions, and you're able to look at different genes. So specifically for our research, we looked at genes related to metabolism, genetic information processing, environmental information processing, and cellular processes. And so what we found when we looked at the wild type to CF versus CF to CF, so this is change in the immune system, that of the top 10 most significantly different KEG features, or bacterial predicted functions, that 8 of them were upregulated when we gave CF mice a wild-type immune system. And of these 8, these were mainly involved in metabolism, as well as like environmental information processing. But the 2 that were downregulated when we gave CF a wild-type immune system was cation proton antiporters, which are linked to increased bacterial virulence, as well as improved survival in varying pHs. And that's important because the CF intestine has a very acidic pH compared to people without CF. And then the pyruvate oxidoreductases, which are also linked to increased virulence, and they are prevalent in bacteria isolated from an inflamed gut. So by altering the immune system, we, one, not only upregulated predicted features involved in metabolism, but we downregulated predicted features that were linked to increased bacterial virulence. So overall, we know that bacterial dysbiosis in the gut has been linked to intestinal pathologies. We see that the loss of CFTR on our myeloid lineage immune cells is able to influence the gut taxa, making these mice have a less proportionality in the taxa, as well as a phylogenetically more similar taxa to our pan-CF mice. By altering the immune system using bone marrow transplantation, we see that the small intestine is most susceptible to changes. And using KEG pathway annotation, we are able to see that by altering the immune system, we can influence bacterial predicted functions, such as decreasing bacterial virulence features. So overall, the microbiota is influenced by CFTR in the epithelial and the immune system, and that the immune defect is a contributor to the CF intestinal disease. It is not only an epithelial defect. And so with that, I'd like to acknowledge my lab, my PI, Dr. Goshan Wong, as well as the Microbial Genomics Resource Group, and then our funding. And with that, I will take questions.
Thanks, Callie. So there's a common question here from Ben Kopp and Luke Hoffman relating to did your models receive the same diets, water, and other treatments?
Yes, everything was normalized across our mice. Because CF mice typically suffer from intestinal obstruction, they have to be on laxative water, so all the other mice were also on laxative water. They were also co-housed together so that any handling or anything within the cage, all of them got exposed to it.
And then do you know, does the diet influence the immune gut macrophage? Have you looked into this and like effects of high-fat diet?
We haven't looked at that specifically with our mice. Our focus is more specifically on the immune system. And I feel like diet would contribute to that, but because of the fact that we try to normalize everything, again, the wild-type mice would also be on this high-fat diet. So it'd be something that we'd have to look into, but I don't know if it directly relates to this project. But good question.
So also from Luke Hoffman, do you know how the different models differ in terms of intestinal motility or malabsorption?
So that is not something that we looked at as far as intestinal motility. We do know that our CF mice do have a slower motility. We did look at histological changes, and we actually found that our immune knockouts also had increased goblet cell numbers, which mimicked what was seen in the CF. So they may potentially have some intestinal transit changes, but that's not something that we looked at specifically.
And did you measure total bacterial loads in the different models, in the small intestine in particular?
No, everything was like relative abundance using just 16S.
So Sebastian has a question. Do you know how the nutrients present in the diet, again, can modulate the function of immune cells in the CF gut, and how this might impact the gut microbiota composition?
Lots of diet questions. We did not look at anything dietarily. We were really just focusing on the innate taxa that are present, but that would be something that we could potentially look into. We are more of an immune lab rather than a nutritional lab. So
Kelly, I have one more question. So the most important thing that I'm taking away from this, apart from the GI effect, is that it appears that the microbiome effects are cell-specific. And that made me think, what about the site-specific? You know, it's a more focused point, but for example, what would be the difference once you figured out the macrophage part between an alveolar macrophage, an interstitial macrophage, you know, a recruited macrophage to the airway? Mm-hmm. As an example, it could be any other organ. Is that something that you guys are thinking to look into the future? Do you have any thoughts on how that would go?
that's a really great thought. We have not looked at that. The intestinal aspect is really interesting because you have predominantly B cells and T cells in the lamina propria. And then you have the intraepithelial lymphocytes, which have more of like your macrophages and your neutrophils, and then you have neutrophilic influx actually into the intestinal lumen. But we have not looked at specifically like the different subsets of that. I think it would be challenging, but I think it would be interesting. But the fact that we didn't see super differences in like our immune knockouts, we saw some, but not as prevalent as like the whole CFTR knockout. I don't know if it would be as advancing as it may seem.
All right, so why don't we save, Rebecca, we got to your question, but let's just save it for the end if that's okay, so that we can keep moving.
What? Yeah, I gotta.
Oh,
Okay,
no, she has to leave
I do
early,
have
so
to
we
leave
have to go through
early.
that question. So, all right, so Rebecca Valls asks, really great talk. How do your CFTR mutants in different cell types change their natural function? Also, any plans to do metabolomics to confirm some of your PyCross results?
So the lab, I personally have not done this data, but other people in my lab have looked at our immune functions in our models, and we do see that the loss of CFTR on, say, our macrophages and our neutrophils result in decreased bacterial killing, decreased phagocytosis. So overall, removing the CFTR affects their natural function. As for the metabolomics, we would like to do that. I don't know if that's something feasible in my PhD. I would like to graduate sometime, but it is something that we would like to consider. So if you'd like to collaborate, send me an email. As for everyone else, I will not be able to be at the end. So again, you can shoot me an email, find me on LinkedIn, do whatever. I'd love to talk and answer any questions that we didn't get to.
Thank you, Callie, for your excellent work.
Thank you for having me.
All right. Sebastian Riquelme, who's assistant professor at Columbia.
Okay, so here we go. So first of all, thanks very much for being here. Also, thanks very much for the organizers for inviting to participate here. Today we're going to be talking about tolerance and metabolism. Okay, I will try to put in your mind a concept about how metabolism is important in the regulation of the immune response. Okay, and how this can be relevant in cystic fibrosis. So just as a reminder, as an immunologist, we like the word tolerance. So I will go through what is tolerance. So we all know that when a pathogen infects a tissue, this pathogen can promote tissue damage, okay, or organ damage. And most of this damage is promoted by the self-inflammatory response against the pathogen. So if this inflammatory damage is not adequately regulated, this can compromise host health. So the organism, the host, activates a mechanism that is called tolerance, in which the main strategy to protect the organ is by reducing the intensity of this inflammatory response to the infection. But the cool thing is that this regulation of inflammation has nothing to do with the elimination of the pathogens. So at the end of the day, the pathogen is not eradicated, and the host can tolerate the infection because it's suppressing the damage produced by the pathogen. Now, when this happens, the pathogen and the host comes into disagreement in where there is an establishment of a host pathogen coexistence. And this is something that we know as a chronic or a long-term infection. And this is something that happens in cystic fibrosis and in COPD, for example, where we see that there are patients that are chronically infected for years, and the immune cells, they are fine, they are okay with the pathogen, even if the pathogen is there. And this happens with different pathogens, such as Pseudomonas, TB, or Staph. But however, despite the highly clinical relevance of tolerance to infection, so guess what? We still don't know how tolerance to infection is established. And in my laboratory, we are trying to understand how this is being promoted through the standpoint of metabolism, specifically ketogenesis. Why ketogenesis? Because ketogenesis is a metabolic response to organ stress that participates in the preservation of organ homeostasis during different stress. So how is ketogenesis promoted? So for example, if one of you today didn't have breakfast, the amount of glucose in your blood will be down, will be reduced. And this will promote a massive bioenergetic stress in the different organs in where the mitochondria are not able to produce energy. So if this bioenergetic stress is not adequately regulated, the tissue will suffer from necrosis and inflammation, and this will lead to organ damage. So to cope with this bioenergetic and inflammatory stress, the liver takes up fatty acids and promotes, through a mechanism of fatty acid oxidation, activates a process that is called ketogenesis, which consists in the production of ketone bodies. Probably you already hear about this with the famous ketogenic diets, which we'll go into in the next slide. And these ketone bodies, such as beta-hydroxybutyrate and acetoacetate, they can travel through the bloodstream to the different tissues, and they can suppress directly the inflammatory response of the immune cells, and at the same time, they can alleviate the bioenergetic stress by fueling or by stimulating the ability of the mitochondria in that tissue to produce energy. And like that, ketogenesis, through the production of ketone bodies, suppress or limits the organ damage produced during this stress. So because of the relevant ability or the role of ketogenesis in preserving organ homeostasis, we in my laboratory were working in trying to understand if probably ketogenesis is also associated to the establishment of tolerance to infection and to protect the organs from the inflammatory damage during infection. So the two questions that we will be answering are, how ketogenesis regulates tolerance to Pseudomonas infection? First question. And the second one, can ketogenesis affect the ability of Pseudomonas to cause inflammatory airway disease? And also, can this pathogen exploit ketogenesis to limit the damage in the airway, in the lung, so like that the host can tolerate the infection and you can promote a host pathogen coexistence for long term? So to answer the first question, we did the simplest experiment that you can do in a lab, in where we induce ketogenesis in animals by using a ketogenic diet. So to induce a ketogenic diet, what we did is that we fed animals with a diet that consists around 90% of the calories in this diet are fatty acids. In contrast, our control animals, they receive a diet that is rich in carbohydrates, in where around 80% of the calories there are carbohydrates. And then, these animals, we fed them with 1 week. We didn't see any difference in weight, in animal weight, anything like that. And then what we did is that we exposed or not this group of mice to Pseudomonas aeruginosa isolate that we recovered from the lung of chronically infected cystic fibrosis subjects, that we know that they are tolerant to infection. So just to let you know how we collected this isolate, during 10 years around, we collected different strains for these patients. And even if the different strains that we collected, they had different phenotypes, such as a mucoid phenotype or a small colony variant phenotype, you have to believe me here that all of these isolate by separate, they induce exactly the same type of blunt immune response. Okay? They don't trigger inflammation as in comparison with the typical laboratory strains, for example, like PO1 or PA14. So after that, we confirmed that all of them, they trigger exactly the same type of immune response. We decided to use them all together as a mixture, okay? Just as a population of bacteria. So we use this bacteria, we infect these animals with these 17 isolates, and we infect these two groups of animals, and then we analyze how that infection modulated activation of ketogenesis in the host and how that was related with the activation of inflammation in the airway. So as I told you before, tolerance is associated with the decrease of the inflammatory response, but without changing the levels of the pathogen in the tissue. And this is what we observe, is that when you treat this animal with a ketogenic diet, we don't see any changes in the Pseudomonas burden in the airway, in both in the lung and in the bowel. Okay, so everything that I will show you now has nothing to do with the pathogen burden. It's only regulation of the immune response. So as expected, when we analyze the accumulation of ketone bodies like beta-hydroxybutyrate that are being released by the liver, we observe that the infection itself producing increase of these ketone bodies in the bloodstream, but the ketogenic diet increases it even more, which is what we are expecting. And of course, the ketone bodies, they are traveling to the airway, because when we do metabolomics in airway, in bowel from this animal, we see an increase in these ketone bodies as well. Now, as we're expecting as these metabolites are anti-inflammatory, we're expecting that the immune response will be reduced. And this is what we observe. We observe that, for example, we observe that the mice under ketogenesis, they accumulate much less of the different cytokines that we know that can promote pulmonary damage, such as IL-6, TNF-alpha, and IL-12p70. But not only that, we also observe that other cytokines that participate in the recruitment of phagocytes that we know that damage the lungs, such as neutrophils, and that also that promote their activation, such as GCSF or MIB1 beta, they were also reduced. And of course, the amount of neutrophils that were recruited to fight infection was significantly less, indicating that we're answering our first question, that the metabolic state ketogenesis in the host facilitate the establishment of tolerance to Pseudomonas infection in the airway. So now, after said that, we wonder, how is ketogenesis relevant to the CF lung tolerance to P. aeruginosa? And we know that these organisms, they are evolving in the airway of the CF patient along the time. So probably our hypothesis is that maybe this pathogen, they evolve to exploit ketogenesis to reduce the local inflammatory response. So now the host can tolerate the infection, and now the pathogen can exploit that environment to coexist with the host for long term. So to analyze how these clinical isolates of Pseudomonas exploit the ketogenic response, we compared the type of ketogenesis that they induce with the classically used laboratory strain PO1 that we know is not really well tolerated by the host because it promotes a massive inflammatory response that kills the host right away in 24 hours. So for this experiment, we fed animals with a regular show that we know that doesn't induce ketogenesis. We infect these animals intranasally with our collection of Pseudomonas isolate again, and we use PO1. And this is what we observe, that when we analyze the levels of beta-hydroxybutyrate in blood, which is derived from the liver, we got this surprise that the clinical isolate, they didn't induce a massive ketogenic response in comparison with PO1, an hepatic ketogenic response. And with this, we said, okay, so maybe now the accumulation of ketone bodies will be reduced in the airway because now the liver is producing less, so maybe there will be less accumulation of these ketone bodies in the airway. However, when we went to perform metabolomics in the airway, we observed that these clinical isolate of Pseudomonas, they induce exactly the same levels of beta-hydroxybutyrate in the airway. Suggesting that probably they are not, the host is not responding against their infection by inducing an hepatic ketogenesis, but probably the host is responding by activating a local source of ketone bodies. And this was super interesting for us because this will be like one of the first signs that we are seeing that there are some pathogens that they are triggering a different type of ketogenesis in the host. So the big question that now we have in my lab is, what is the airway source of ketogenesis that these clinical isolates of Pseudomonas are exploiting? And how is this relevant to cystic fibrosis? So to analyze what is the pathway that is associated to induce this local production of ketone bodies, we decided to analyze the entire airway metabolome of mice infected with this collection of clinical isolates. And what we observe is that when we analyze the different metabolites, we observe that in addition to be the most accumulated metabolites in the airway of these mice, these ketone bodies, they cluster with itaconate. And maybe you have heard about this metabolite. It's a super famous metabolite in the field of immunometabolism. And the important thing about itaconate is that it's an anti-inflammatory metabolite mainly produced by myeloid cells. And at the same time, it's a natural bactericidal molecule. But recently we published that Pseudomonas doesn't succumb to the presence of itaconate, but indeed can exploit itaconate to produce biofilms. So why not? Maybe these clinical isolates, they have evolved in the airway of the CF patient to exploit the production of itaconate to trigger this accumulation of ketone bodies. And we were expecting that this was not going to be the case with PO1, because PO1 is not adapted to the host. And we observed that even if we observed the 3 different metabolites that were accumulated in the airway, they didn't cluster in terms of abundance. So maybe this corroborates our idea that maybe itaconate is associated with the local production of ketone bodies with these clinical isolates. So to confirm to answer that question, what we did is that we took animals that can produce itaconate or that cannot produce itaconate because they lack RG1, which is the gene that produces itaconate, and we challenge them, we infect them with PO1 or the Pseudomonas isolates, and then by metabolomics, we analyze the presence of beta-hydroxybutyrate, this super famous ketone body. And as we were expecting, when the host produced itaconate, now the airway got enriched in this beta-hydroxybutyrate during the infection with the clinical isolate. And this is something that we didn't see with PO1. In fact, I have all the data that I'm not showing you here. These ketone bodies that are coming from here with PO1 are mostly derived from the liver, okay? But these are being accumulated there are not derived from the liver because when we analyze the level of ketone bodies in blood, in mice infected with the clinical isolate, we don't see any difference in the hepatic response, in the hepatic ketogenesis. So this again confirms that these ketone bodies are being produced in situ, in the site of infection. And these pathogens are exploding itaconate to induce that. So to wrap this part, this tells us that the CF pyranose isolate, they exhibit unique metabolic properties in comparison with laboratory strains. So we have to be careful when we work with PO1 or with laboratory strain, or with clinical isolates, because they don't have exactly the same type of, they don't induce exactly the same type of metabolic response and immune response. So is the CFR we're reaching itaconate and beta-hydroxybutyrate? That's the question. So in collaboration with Clemente Brito here, sitting next to me, he shared with us some sputum samples, and we quantify by metabolomics the presence of itaconate and beta-hydroxybutyrate in sputum from CF people. And this is what we observed. We observed that both metabolites were accumulated in the airway of cystic fibrosis patients. And we were wondering, are these ketone bodies being derived from the liver? And to do that, we, in collaboration with Dr. Emily Dimango at Columbia University, we analyzed the presence of the same metabolite, beta-hydroxybutyrate, in bloodstream to see if they are coming from the liver, and we observed that there are no difference. So most probably, these ketone bodies in the CFR way are being produced locally and potentially through the action of itaconate that we know that is being exploited by these pathogens. So just to finish, this is the last part. So how the ketone bodies and how the itaconate are being accumulated in the CFR way are reducing the ability of pseudomonas to promote inflammation. So the hypothesis that we have is that potentially these anti-inflammatory metabolites, they are affecting the ability of pseudomonas to produce LPS in the surface, because we know that LPS is the pro-inflammatory molecule that is present in the surface of these pathogens in gram-negative. And maybe you know this, but to expose LPS on the surface, pseudomonas utilize a transporter that is called LPTD. And LPTD, what it does is that it mobilizes the LPS that is already a mature LPS from the periplasmic space to the surface of the bacteria. And when the LPS is there, now the LPS can be sensed by the immune cells or by epithelial cells and that trigger the inflammatory response. So the hypothesis is that probably itaconate ketone bodies, they were suppressing or limiting the ability of the pathogen to express LPTD. So we did the ECS experiment as well, again in the laboratory, in where we exposed PO1, which is a laboratory that has never seen the airway, and we observed that just by stimulating this pathogen with itaconate or beta-hydroxybutyrate or acetoacetate or both ketone bodies together, the levels of LPTD were going down. And this is something that is really happening in the CF airway, because when we analyze the LPTD levels in the Pseudomonas isolate, we observed that, for example, in two representative isolates from our collection, LPTD levels are really low in comparison with PO1. And not only that, but also in another collaboration with Dr. Barbara Colt, I think he's here in the audience, she shared with us also other a collection of longitudinal connection of Pseudomonas aeruginosa from another chronically infected tolerant CF patient. And we observed that this isolate also, they have less level of LPTD in comparison with PO1. So you think that these changes that are being promoted by these anti-inflammatory metabolites are really happening in the CF airway. And probably you don't believe me that by losing the LPTD, the bacteria, they become less inflammatory. So we did the other experiment, is that we remove LPTD from PO1 and we infect animals. And of course, these animals, they accumulate less pro-inflammatory cytokines that we know that damage the airway tissue, such as IL-1 beta, IL-6, and TNF-alpha. And of course, this was exactly the same pattern of cytokines that we observed with our collection of clinical isolate, that we know that they lack LPTD in bacteria and they don't expose LPS. So to finish, ongoing conclusions. How is tolerance to infection established? We think that tolerance to infection is established by the activation of ketogenesis, this major metabolic pathway that is promoting the host when we have a bioenergetic stress in our tissues, or also when we have an inflammatory stress in our tissues. And now, with what we think that is going on is that Pseudomonas aeruginosa, it's exploiting this ketogenic response by inducing itaconate in the airway, and this is promoting pathogen adaptation, and like that now the pathogen exposes less LPS in the surface, and that helps them to mitigate the inflammatory response in the airway, and like that, now the host can tolerate better the infection, and now the host and the pathogen, they can coexist for long term, and that is what we call a chronic infection. So, to finish, I would like to thank my laboratory, and also would like to thank the laboratory of Dr. Ali Prince, which we maintained like in collaboration. And most of this work, I did it in collaboration with Kira Tomlinson, that she already gave a presentation about itaconate. I would like to also thank our collaborators, the funding, and if there is someone in the audience interested in working in immunometabolism and the regulation of inflammation through metabolism, I have a postdoc position available in my laboratory. So if you're interested, you're welcome to come to talk to me. So thanks very much.
So Katie asked, regarding the first experiments, ketogenic diet lowers the amount of CO2 that mammals need to excrete this, and this changes respiratory rate and or work of breathing. Does ketogenic diet affect pH in the airway?
We haven't tested yet, but that is a super good point, and you're right. Well, we haven't tested yet.
Hassan wants to know if the LTBP knockout PaO1 remain longer in the lungs of infected mice.
Okay, that bacteria remains a couple of days and then is eradicated. And it's because that doesn't necessarily replicate exactly the phenotype with the clinical isolate, because the clinical isolate, they have also other adaptations that we also see that are happening when you expose these bacteria for long-term to itaconate. For example, you have increase in extracellular polysaccharides like alginate that really help the bacteria to stick longer in the airway.
All right, so hear me out on this crazy question. So one of the most powerful inhibitors of ketogenesis is insulin, right? Clinically and physiologically. So do you see a relationship between these patients that develop CFRD, have minimal insulin expression in their terminal stage and their increased prevalence of Pseudomonas? And then the question that follows is there a role for us, is there a future in which we would give a microdose of insulin for an infection control issue rather than a metabolic management issue?
I mean, I will start from the last one. I think it's, yeah, why not? We are just starting to this. The idea is that as we know how important metabolism is the regulation of the inflammatory response, why we cannot take advantage of that to regulate through the insulin administration and also through the diet, why not? We can do all of that. Or probably also we are doing other experiments in the lab where we are trying to administer directly these ketone bodies to the airway of mice. Why not? Okay, maybe that can be nebulized, we don't know. We're just working on that.
So people are getting excited about asking questions. So they all came at once. So beside ketone bodies, are there other molecules you would expect from catabolism induced in the airways by infection by CF isolates?
All the type of metabolites that could be doing the same, that's the question?
Are there other molecules that you would expect from an infection that induces a catabolic state in the airway?
Oh yeah, there are other, yeah. There are, I am not working with that, but I know that there is like accumulation of some short fatty acid that they are kind of similar. For example, acetate, you have propionate, that also they are related with the activity of the microbiota. Also, they can do something. We have some other information that I don't want to share too much, but there are other metabolites, other metabolic pathways that are related. But it's related with the, I will say anyway. So the idea is that everything, what's triggered, what we have learned from the world of immunometabolism is that when you screw up with the mitochondria in immune cells, the cells, they cannot produce energy and they activate compensatory pathways that exacerbate inflammatory response, such as glucose, okay? So that's why we see that the immune cells, they activate glycolysis. So the idea is that the host respond to that metabolic stress by trying to wake up the mitochondria to produce more energy. So the hypothesis that I'm working in my laboratory is that probably a good idea will be to come and to rescue the ability of the mitochondria to keep doing energy. And like that, the cell will compensate back and will reduce the pro-inflammatory glycolysis. And when I'm talking about restoring the ability for the mitochondria to produce energy, I'm talking about anerplerosis. It's a super weird name. But anerplerosis is the way how the cells can restitute the ability of the TCA cycle in the mitochondria to produce energy. And who promotes that? Amino acids like glutamine, you have glutamate, and also fatty acids that enter into the TCA cycle to fuel the ability of the mitochondria to keep producing energy.
So I just want to state for the record that Lucas Hoffman's question made Sebastian Riquelme give up the game. Moving up to, so one more question, and then we'll have a couple left over at the end. Megan Hirsch asked, so she says, great talk. Did you by chance look at the levels of iolate? If so, did you see the same effect with the keto diet and Pseudomonas given iolate's role as a neutrophil recruiter?
Honestly, I don't remember if we have iolate in our panel. I think we do. And I think it should be down because around the 90% of the cytokines were down. So I can recheck and I can say it. I don't want to lie.
All right, so we'll leave the other three questions that we have here for the end. Thank you so much, Sebastian.
Thank you very much.
Work. Thank you so much.
Okay. Our next speaker is Nicola Lohr. He's going to talk about IL-17.
So, before to start, I would like to thank the scientific committee to give me this opportunity to present our work focused on the role of IL-17 receptor during chronic respiratory infection by Pseudomonas aeruginosa.
You can go with the arrows.
Yeah. Okay, right. Yeah, this is my disclosure. So, the exaggerated inflammation and tissue damage associated with the chronic persistence of Pseudomonas aeruginosa are, as already described, a central pathological feature of the chronic respiratory disease in cystic fibrosis patient. And the interleukin-17 inflammatory pathway has been widely described as a key player in cystic fibrosis pulmonary disease. Also recently, in particular in work where IL-17A and TNF can act together to improve the effect of modulators. So in general, it has been widely described that IL-17A and IL-17 receptor A axis can play a double sword activity, in particular in response to Pseudomonas aeruginosa. From one hand, they can orchestrate the infection, the inflammation at the early phase of the infection, and from the other, they can modulate the immunopathology that occur during chronic infection and the tissue damage associated. So it's worth noting, going details, that the IL-17A and the IL-17 receptor A axis, it has been widely described. The IL-17 cytokine A and F can act through the heterodimer formed by the IL-17 receptor A and the IL-17 receptor C, activating several downstream pathways associated with pro-inflammatory cytokine, antimicrobial peptides, mucins, or tissue damage remodeling factors. It's worth noting that IL-17 receptor A can act in a heterodimer form, not only activating pathway mediated by IL-17A and F, but also activating pathway by IL-17E and IL-17B that can activate downstream pathway completely different than those promoted by IL-17 receptor A and IL-17 receptor C. And in addition, this receptor can also form another heterodimer activating pathway mediated by IL-17 receptor C. So, although in the past much of the attention were focused on the IL-17 receptor A, in our work we were interested to understand in particular what are the role of this receptor in modulating the host resistance against Pseudomonas aeruginosa infection, in particular exploiting chronic respiratory infection by this pathogen. So to try to achieve the final goal of our project, the first question was to determine the level of these cytokines during chronic infection by Pseudomonas aeruginosa. And to do this, we took advantage of our model of chronic infection, where we can recapitulate the early phase or acute phase of the infection, such as at 2 days post-challenge, and the long-term chronic infection and associated immunopathology as observed at 28 days post-infection. So we used this model and the agar-bits mouse model, and we exploited the cystic fibrosis clinical strain named A43. And in this model, we characterized the expression of the IL-17 cytokines and receptor at the early phase and advanced stage of the disease, of the infection process. So in this model, we confirmed the data that we previously published, so that IL-17A and F are activated at 2 days post-infection and further released after 28 days post-infection. Then we looked to the other cytokine, and we observed that IL-17B and IL-17E were also upregulated during the development of chronic infection, and a similar trend was observed for IL-17C. So overall, this data, and in addition to data that I have not the time to show here, but we checked also the expression of the receptor within the lung compartment between stromal inflammatory cells, confirmed that this pathway, this axis, are activated during the development of the chronic infection. So the next question that we wanted to address was in particular how we can selectively target the contribution of this receptor on host resistance during chronic infection by Pseudomonas aeruginosa. And to do this, we decide to develop 3 new mouse models through a CRISPR-Cas9 technology. We selectively depleted IL-17 receptor B, IL-17 receptor C and CFTR receptor E in the same, in mouse displaying the same genetic background. This was an advantage because it gave us the opportunity to compare the host response in the same experimental settings, rather than to compare this response using mouse that were previously generated, but with a different genetic background, with a different genome editing approach. So we confirmed, that this receptor were not expressed in this new mouse model, and we exploited these 3 new mouse model in our agar-bits model. And again, here we evaluated the bacterial burden at the early phase of the infection and the advanced stage of chronic infection, the incidence of chronic infection, and the overall leukocytes recruitment in terms of CD45 positive cells. And focusing on the early stage of infection, we found out the only IL-17 receptor C knockout challenge with Pseudomonas aeruginosa display a higher bacterial burden in comparison to the control infected mice. In terms of CD45 cells, we didn't observe any difference. And this was partially in line with previous publication that described in the early phase the role of IL-17A and IL-17 receptor A. Going ahead, we dissected the bacterial burden at 28 days post-infection, and we confirmed that only the IL-17 receptor C knockout mice display a significantly higher bacterial burden in comparison to the control group. And again, we didn't observe any further increase, overall further increase of CD45 cells in comparison to the control. Moreover, another interesting thing that we observed was the incidence of chronic infection, and we found out that the lack of IL-17 receptor C significantly increased the susceptibility of these mice to Pseudomonas aeruginosa chronic infection in comparison to the wild type 1. So, based on these results, we confirmed that, or we observed that IL-17 receptor C was the only receptor involved in host resistance, and so the next question was to address the role of IL-17 receptor C in modulating lung innate and adaptive immunity during chronic infection, and in particular, we focus, recapitulate the experiment with the agar-bits model at 2 days and 28 days. In this case, we want to focus our attention on the inflammatory response, and in particular in the bronchoalveolar lavage fluid and in the overall lung. We dissected innate and adaptive inflammatory cells, and in this talk, I'm going to focus my attention on the advanced stage of chronic infection. So at 28 days, we found out in bronchoalveolar lavage fluid that the mice lacking IL-17 receptor C displayed higher monocytes and neutrophilic recruitment in comparison to the mice, to the wild-type mice infected with the CF clinical strains. And this trend was also confirmed in terms of cytokines, where Kc and MIP2 were tends to be significantly higher in the airways of mice infected with AA43 and lacking the IL-17 receptor C receptor. We go ahead and we wanted to evaluate the cytokines in the overall lung homogenates, and we found out that cytokines typically involved in the type 17 immunity were upregulated, in particular, as in this case, Kc, GCSF, IL-6, or IL-5. So we go ahead and we wanted to further characterize the adaptive immune response. And we didn't observe any difference in terms of burdens of CD3 and CD4, but when we started to characterize CD4, we started to obtain some difference in terms of phenotype. We wanted to characterize CD4 secreting IL-17 as Th17-like cells, or CD4 secreting interferon gamma as Th1, and those secreting IL-4 as Th2. And we found out that the Th17-like cells were significantly higher in the mice infected with the CF clinical strain and lacking the IL-17 receptor C IL-17 receptor. We didn't observe this variation in Th1-like cells, although with a slight increase of these cells, and we didn't observe any variation in the Th2 response. So suggesting that overall, the lack of IL-17 receptor C seems to exacerbate type 17 immunity through the exacerbation of CD4 IL-17 positive cells and an increased recruitment of neutrophils into the airways. So to confirm this data, we were interested to localize these cells, in particular this Th17, in the inflamed area. So to do this, we performed Visium spatial transcriptomics in order to determine whether the T helper cells were located in the inflamed or non-inflamed area. And with this technique, we can compare the transcriptomic profile in the spatial tissue. So we can compare the transcriptomic profile within hematoxylin, within lung tissue stained with hematoxylin and eosin. So as you can see here, each dot of this picture stained with hematoxylin and eosin represented the transcriptomic profile of around 20,000 genes. And thanks to data set already published related to Th1 and Th17, and in particular those published by our collaborator Chen Kong from Pittsburgh University, we were able to deconvolute our data set and identify the Th1 transcriptomic profile and the Th17 transcriptomic profile. And as you can see here, we confirmed that wild-type mice do not have a higher level of Th1 transcriptomic profile, as represented by the increased intensity of the red color, while Th17 are still present, and in particular in the inflamed area. When we observed the knockout mice infected with A43, we also confirmed that Th1 were not mainly involved, but Th17 were strongly involved in the inflamed area of these mice, so confirming the data observed by FACS analysis. And we quantify, of course, this data, and we found out that a slight increase of Th17 was observed in mice lacking IL-17 receptor C, while the Th17 profile were activated in the wild-type mice, but were strongly activated in the IL-17 RC knockout mice. So up to now, we can conclude that during the development of chronic respiratory infection, we found out that all the IL-17 cytokines are upregulated, and we confirmed also the presence of the receptor between stromal and non-stromal cells. In addition, we found out that only the lack of IL-17 receptor C, but not IL-17B or E, impairs source resistance. And we also found out that IL-17 receptor C, the block or the lack of IL-17 receptor C, upregulated a pro-inflammatory response mainly mediated by a type 17 immunity and characterized by the recruitment of neutrophilic recruitment in the airways and Th-like 17 cells in the inflamed area. So before to conclude, I would like to thank all the collaborators. In particular, I would like to thank the people of the Emerging Pathogen Unit working in this group. I would like to thank also the collaborator at the San Raffaele, Ospedale San Raffaele that helped me in this project. Our external collaborator at the University of Pittsburgh, Chen Kong, and the Cystic Fibrosis Foundation that provide us funds to support this work. I thank you for your attention. I'm ready for your question.
So we have a question. Did you check the levels of TGF beta in the IL-17C knockout mice at 28 days of chronic infection?
It's something that we didn't check the TGF. We checked for remodeling process, and we didn't observe a greater remodeling process. It's something that we want to confirm also in terms of molecular pathway, but for example, we didn't observe any difference in terms of T regulatory cells, so we believe that are not so, this should not be so much involved in our, at least in our model.
So, so what do we know about people going on to modulators? Do we know what's going on with their Th17 response? Is there any data?
Okay, there's a paper showing that the cytokine IL-17A and TNF can in some way improve the activation, the function of evapcaptor, and so in some way there's a discussion related to the fact that probably IL-17A, as in the past has been suggested as a potential target to limit inflammation, may be, a little bit harmful to block, and so in some way also to limit the action of this modulator in vivo. Another thing that it has been described, I guess, in terms of generic pro-inflammatory pathway in the blood, was a reduction of the Th17 response in people that get a modulator, although these are really preliminary study and it's really hard to speculate or to conclude at this moment.
There's another question here, a comment. Despite 4 weeks of increased bacteria in the lungs of your IL-17 receptor C knockout mice, you didn't see any more CD45 immune cells in the total lung, but you did see more innate immune cells in the BAL. Can you comment on this observation?
Yeah, okay. The CD45 cells were quantified by FACS in the overall lung, so that includes a huge amount of cells that then were differentiated in the fraction that we evaluated. It's worth noting that the bronchoalveolar lavage fluid were evaluated by diffuse staining because the FACS analysis of those cells in the infected mice, it's not easy, and the only way to evaluate it by FACS is combine much more mice together, and this was a limit, in our opinion, for our results. Moreover, the other things that we differently observe were the Th17, although in terms of expression, so there were a number of CD3 and CD4 were not different. So we guess that the variation that we didn't observe in CD45 is due to the fact that CD45 cells include a so huge amount of cells that, in our model of chronic infection are not only represented only by neutrophils.
So, Nicola, in your spatial transcriptomics data, did you see, so is that difference in IL-17 expression, is it uniform, or is there a gradient towards a particular cluster of cells or infection?
Okay, we didn't perform single-cell analysis to deconvolute directly the sample because it's really tough, in our opinion, to combine single-cell with spatial transcriptomics in our sample because the inflammation is really local. And so when you perform single-cell RNA-seq analysis, and you usually seek like 3,000 cells for sample, you risk to, in some way, to not represent the real composition of your tissue. We took this strategy to get the single-cell data obtained from the University of Pittsburgh and try to deconvolute this for this main reason. And we observed that although also in the non-inflamed area, I'm not going to show here because it was a little bit tough to also to explain in so little time, the inflamed area have a sort of density wave of activation of the signaling. So it means that much more closer are the dots to the inflamed area, much more higher is the signaling associated with Th17. So I guess that this is a really cool technique in order to evaluate better the inflammatory area associated to the bacterial infection and the presence of the bacteria. So in the next time, we want to characterize the localization of the bacteria and then associate the inflammatory process around the bacteria.
Yeah, we're looking forward to hearing more about that. Well, thank you so much for your outstanding work, Nicola.
Thank you.
Okay, our last speaker is Hassan Oz from Yale.
Right. Thank you for the kind introduction, and I'm very happy here. And I want to thank the organizers for saving best for last, and I want to thank all of you for sticking out here with us. And yeah, today I want to talk about how the impaired recruitment of CCR2 positive monocytes does not impair the host defense against pulmonary Pseudomonas aeruginosa infections. I have nothing to disclose. So as we've heard several times over and over again, CF lung disease starts early in the life of CF patients and is characterized by mucus plugs, thick and smooth muscle airways, and a cycle of inflammation and infections that over time damages the lungs. But what I want to point out here is in the late-stage disease, we really have damage that reaches out into the alveolar spaces of the patients, as we see in lung explants. And together with that, we also was shown in 2013 already by the group of Dr. Harris, that the lung explants of CF patients have abundant amounts of collagen depositions, and that is triggered by TGF-beta signaling, which is shown here with phospho-SMAD2 levels. We also had our go at lung explants that were provided to us by Dr. Randell and the UNC Tissue Procurement Center. And we looked at the immune cells around the airways, and we saw that around the airways in the submucosal area, we have abundant amount of CCR2 positive cells. And in a study that Dr. Brito did in 2020 and at Yale, and they showed a single-cell RNA-seq study from sputum of CF samples that monocytes are very abundant in the sputum of CF samples, while the healthy controls mostly had alveolar macrophages. We asked them kindly to have a look at the data again and to see whether these monocytes also express CCR2, and in fact, they do express CCR2. So that was very interesting to us. So we had the confirmation that these cells are there in the lungs of CF patients at late stages and also in the sputa. So talking about lung macrophage and monocyte populations, this is a very simplified version. We have two major populations, the interstitial macrophages and the alveolar macrophages. And upon an inflammatory insult, for example, an infection, we have recruitment from the circulation by classical monocytes. These are CCR2 positive, and they are recruited via CCR2. And in the mice, they are Ly6C positive. They are in humans known as CD14 positive classical monocytes. And they then come recruit to the lung. They help out with the infection to mount a pro-inflammatory response. And they can then later on differentiate into interstitial populations, alveolar populations, and overall help to calm down the inflammation. Now, we wanted to study these, and what we do is we used mouse models. So we have wild-type and CF knockout mice. And we also wanted to, of course, look at the recruitment of these cells. So we used CCR2-deficient knockout mice. And to have a look at the CF background, we also crossed these mice with our CF mice to create CF and CCR2 double knockout mice. And first thing we did was we looked at a chronic inflammation model to really mimic the lung remodeling that we have in the CF patients. And this was already published before, but we repeated the experiments where mice are exposed to LPS for 5 weeks, 3 times a week, and then analyzed or left to recover for additional 6 weeks to see whether they can reinstate the lung homeostasis. And our data from there shows that in the CF mice, especially at the chronic time point, we have huge enlarged alveolar spaces. So the alveolar space is disrupted. And more importantly, at the recovery time point, while the wild-type lungs look fairly normal, the CF mice still remained with enlarged alveolar spaces, so the damage is sustained in the lungs. The double knockout mice, on the other hand, while they did have some damage at the chronic time, in the recovery time they were looking mostly fine. And we had this also quantified by a pathologist, where it showed that the CF mice remained with high damage scores, while the double knockouts were reduced at the chronic and the recovery time. And interestingly, the CCR2 knockout mice, they were completely protected from any lung damage. We had a look at the lung immune cell populations, and there we see that at the chronic time point, compared to wild-type mice, the CF mice had highly increased numbers of classical monocytes, which of course in the deficient mice were highly reduced, and the neutrophils were also increased in the CF mice. But importantly, what I want to point out here is that the double knockout mice that have impaired recruitment of monocytes also had lower amounts of neutrophils in the CF background. They were close to the levels seen in wild type. At the recovery time point, we also see that the increased numbers remained in the CF mice, not as high as in the chronic time point, but it also the same with the neutrophils, while the double knockouts were at lower number of neutrophils and monocytes. From there, we wanted, of course, to know what about TGF-beta levels and lung remodeling, all those things. And we see that the TGF-beta levels were increased in the CF mice in the chronic time point. The double knockouts were lowered to levels similar in wild-type mice. And also the phospho-SMAD2, so the signaling levels, were also increased and lowered accordingly. At the recovery time, we also had more TGF-beta signaling left. We also looked, I'm not showing it here, we also looked at target gene expression like collagen 1, and that had a very similar profile as well. Lastly, what we did here in this study was that we looked at potential inhibitors, and these are small molecules that some of them are already in clinical trials and tested for other disease types. And we gave it to the mice in the drinking water over the time course of the 5 weeks of LPS nebulizations. And CF mice, what happened there was very similar to the knockout mice, although not as severe in the terms of recruitment of monocytes. We have a reduction of the monocyte recruitment that also translated to less neutrophils, less TGF-beta signaling. So overall, we can show that the inhibition of CCR2 also leads to lower TGF-beta levels. So the small summary from this previous study is that recruited monocytes cause lung damage in CF, and the inhibition of classical monocyte recruitments by targeting CCR2 leads to lowered lung damage. But of course, I mean, as you heard before, it's always about the balance. And while in the healthy subjects we have good host defense and good inflammatory responses that are fine, in CF we know that the balance is highly shifted towards a worse defective host defense and hyperinflammatory phenotype that causes a lot of damage. So talking about antimicrobial defense, we wanted to know, of course, can we just target the recruitment of monocytes, or will we have a problem with this? So the question was really, does the inhibition of the recruitment of CCR2 positive monocytes to lung tissues compromise the host defense? And we wanted to look at a major CF pathogen, so we used Pseudomonas aeruginosa. The model is very simple, straightforward. We infected first wild-type and CCR2 knockout mice with 5 × 10⁶ CFUs of PA01. And we looked at several time points throughout. We collected bronchial lavage, lungs, and spleens to look for CFUs and to look for lung immune cell populations. And I want to start with the lung immune cell populations, where we see that within 6 hours of infection, monocytes are recruited to the lungs, and they remain elevated up until 4 days after infection. Of course, the recruitment was deficient in the CCR2 knockout mice that we see throughout the time course. Then when we look at the neutrophils, their recruitment was not impaired. And I think that was an important point for us to see because, of course, they are the ones that are going to fight the bacteria mostly. We did see a slight increase here at 72 hours, but I want to point out that we only have an N of 3 here, so that requires some more repetition. Alveolar macrophage numbers were not altered comparing wild-type to CCR2 knockouts. Now, if we have a look at CFU counts in lung and bowel combined in this graph, we see that both mice start to clear bacteria within 4 days, or lower the amounts heavily, and there was no significant difference comparing CCR2 to wild-type mice. We also had some dissemination to spleens, which we initially did not expect, but was very consistent throughout the experiments, where we see at 24 and 48 hours some spleen bacteria, but they were also cleared throughout the experiments, and there was no difference. So we could say that the clearance of PA01 was not impaired by CCR2 deficiency. But now, of course, coming back to CF. So we added our CF mice and the double knockout mice, and we looked at 24-hour time point for this. And what we observed here is first that, of course, the monocyte numbers were severely impaired in recruitment to the lungs in both wild-type and CF mice in the CF backgrounds, whereas the neutrophil numbers, they had no difference in the wild-type versus CCR2 or CF versus double knockouts. There was a slight increase, but it's not significant here. Alveolar macrophages were not affected at all. And we have a look at the CFUs. We, as kind of expectedly, see that CF mice had higher number of lung CFUs of PA01 at 24 hours post-infection. But again, here, we do not see a difference comparing wild-type to CCR2 or CF to double knockouts in terms of the CFU counts. Spleen, very similar phenotype. We did not have really a difference comparing wild-type to CCR2 or CF to double knockouts, with a slight increase in the overall counts in the CF background at 24 hours. So lastly, we also looked at cytokine levels, just to have a look at what happens in the pro-inflammatory field. And there are three ones that I want to point out here, which is IL-6, interferon gamma, and IL-17, which we just heard about. They were all increased in the CF mice in the bronchial lavage. But again, here, what I want to just repeat myself, the CCR2 deficiency did not affect the cytokine levels. So to summarize everything, I showed you that CCR2 monocyte-derived macrophages drive a pathogenic recruitment of neutrophils and TGF-beta signaling in lungs of CF mice that were exposed to chronic LPS, and that led to irreversible lung damage. And we also showed that blocking their recruitment prevents lung tissue remodeling. And in this study, we showed that impairing the recruitment of CCR2 monocytes does not impair the initial clearance of PA01. However, the later time points we definitely need to assess as well, especially in the CF knockout mice or the double knockouts. And as future directions, we want to look at more clinical isolates. As you heard, they are very different from the PA01, and also look at chronic infection models. And obviously, different CF-relevant microorganisms that need to be assessed. And lastly, a pharmacological approach to target CCR2 instead of the full genetic knockout. And with that, I want to thank everyone who's been involved with this, the Yale CF Center, Dr. Egan, Dr. Murray, who was very helpful, and our infectious disease guru. Dr. Brito for helping with the previous studies and always being a happy soul around. And Dr. Randall for providing us with lung tissues. And of course, my lab, and Dr. Busha for the great support that I have. Thank you.
Right, so Sebastian says, excellent talk. Do you know how CCR2 regulates lung inflammation during infection with pathogens that lack LPS, like staph, for example?
That's a good question. I have looked a little bit into literature, what was done in the wild-type or CCR2 knockout mice. And actually, let me just pull it up here. I have an extra slide because I'm not sure there's a lot of different ones. So for staph, there was actually one study that looks interesting, which was the last one down here. But there was a co-infection with a prior H1N1 infection, where they showed that CCR2 knockout mice had actually better survival. I don't think that there is a direct impact on basically LPS, yes or no. In terms of CCR2 recruitment, I don't think that TLR4 is the major key here, since I think it's more about a general inflammatory response that the monocytes will be recruited also for staph infections.
All right, so next question is your mouse infection an alveolar infection, or do you think you would see different results in CCR2 knockout mice if you use agar beads and small airway infection, which may be more similar to CF?
Small airway infection, okay. So what we did is I think I mentioned, so we did like intratracheal application. So we really intubated the mice and made sure that the bacteria basically reach into the lungs, and it's not just an intranasal, and who knows where the bacteria go. I don't know about agar beads. We did not do anything with them. So that is something I'm always a little bit on the, do I want to go towards the agar beads or not? And I'm trying to just to say, I'm trying to figure out a way to get to a chronic infection without using beads, just to have a little bit more similar phenotype as you would expect in the CF patients. So probably that's why I'm always asking about, do the bacteria stay around longer or not? That was the main idea of that. Yeah, so I'm not sure. Like, yeah, we're using basically a model where we're sure that they reach into the lungs.
Go ahead.
So, do you know, are the recruited monocytes involved in clearing neutrophils? Do you see an accumulation of apoptotic and necrotic neutrophils over your time course?
Oh, that's a good question. So, I mean, that is a good question, yes. Yeah, so monocytes, macrophages, in some way, yes, are definitely there to also clear out neutrophil bodies and do basic some of the efferocytosis cleanup. Whereas in this early acute models, I'm not sure how relevant they are for that, but we definitely see in the chronic models that the alveolar macrophages are definitely there to clean up neutrophils. It could be one of the reasons why we maybe see a slight accumulation of neutrophils, but again, we need to repeat and see if that holds true. But it could be. I'm not sure at this point.
My other question is, have you ever measured cathepsin B levels?
No.
It seems like it'd be a good model to see if it was monocytes.
We did not, yes. I actually wrote it down before that we should probably have a look at that, but did not do that, no.
So, Hassan, two questions. One is, how do you understand the very specific effect on monocyte recruitment, but not on neutrophil recruitment? Do you think they're independent processes? Do you think the CCR2 recruitment process is completely separate from the neutrophil recruitment process in this infection?
Yes, I mean, we see that especially monocytes rely on CCR2 for the initial recruitment to the lungs. They need it for the efficient recruitment. Neutrophils, on the other hand, have other ways. They have other receptors and different cytokines that attract them. So yeah, I would definitely say, like overall, the inflammatory response will try to attract basically both of those populations. But in the CCR2 knockouts, we have the deficient recruitment because they can't respond to the cytokines.
I guess this leads to my clinical question, which is, how do you see the clinical application of this? So it would be cool because
Yes.
it's cool because it's specific, right? So at least you're going to have a neutrophilic response to kill bacteria, even if you reduce the CCR2 response. But is this monoclonal antibody against the CCR2 receptor? Is this a cell therapy? Like, how do you see it happening?
That is a good question. I mean, in clinical trials right now, it's some small molecule inhibitors. They try to go that way, and it's mostly studies where they look in cancer or also in autoinflammatory diseases. So it's not really an active infection or inflammation thing. And yeah, that is actually a very good question. I'm at this point not sure how to really go about it. If you would want to go when there's an exacerbation and then maybe go at that point to limit the further recruitment of the monocytes, or as a baseline therapy, I don't think that would probably be very good, since monocytes are needed for other infections. So I think it's probably going to be something more towards a guided or directed time point where maybe there is a immediate exacerbation, and then we try to limit the number of monocytes getting recruited. But at the same time, yeah, it's something, this is very basic. I just wanted to see in the beginning, does it affect it at all in mice or not? Because if it did, then it would be done already, right?
All right, well, thank you so much for your outstanding work, Hassan. Thank you.
Thank you.
All right, and we have a couple of leftover questions that we can go through. So one was a question for Sebastian from Diana Celedonio. She wanted to know if your clinical isolates had LPTD mutations in them. Yeah, come up.
So that's a great question, and yes, they have a lot of mutations. We did genome sequencing of all the isolates, and they do have a lot of mutations, non-synonymous mutations mainly, but also they have some synonymous mutations, so they do.
And then the last question for you too, Sebastian, from Katie. Could you comment on commonly used cell culture media glucose concentrations like RPMI and DMEM, which are all hyperglycemic? Are we not using the correct media to study immune cells in the lab?
Maybe yes, we are using the wrong media. Because some of the media, they have more than 5 millimolar glucose, and you are in the around diabetic, range, and that can even potentiate even more the inflammatory response of cells. So it would be optimal to use other type of media, but I know that it's complicated because cells in culture, they don't like it, so we are in a problem. So I recommend you to do in vivo models.
All right, thank you, Sebastian. So I want to, with Matt, we would both like to thank you all very much for sticking it out until the very, very end, for your brilliant questions, for all the presenters, for their outstanding work, and also to our patients that contributed to this work and that are always cheering us on to keep going. So thank you very much, and I hope that we see you next year.